This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, Episode 5 for October 21st, 2008. I'm Vincent Racaniello, and I'm here with a guest today, Saul Silverstein, who has been a colleague of mine at Columbia for more than 25 years?
About.
Dick Despommier is not with us this week. He's off to PopTech, which is a conference up in Maine, to promote vertical farming. So we're here to talk about herpesviruses today. Saul is the expert virologist. Now, in contrast to Dick, who is not a virologist, Saul is a virologist, more so than I am, far more intelligent and learned than I am. So it's great to have him today. So we're going to talk about herpesviruses, which is his specialty. Isn't that right?
We are certainly interested in them. They are fascinating little beasts.
All right. So we'll have plenty to say about herpesviruses. But before we do, Dick passed along to me several articles that he found in various Indian newspapers yesterday. Last time we were talking about rabies and rabies virus. And it turns out that rabies is a huge problem in India. Did you know that?
Yeah. There are lots of different sources of rabies, and we keep finding new ones, and one always looks at the sources of resources such as bats, which can be almost anywhere. Right. They're a constant
bats are a huge source.
Provider of new infections.
Well, it turns out that in India, they have 25 million stray dogs, which contributes to 96% of the country's rabies. Through 30,000 people die of rabies each year in India. And the main problem is they can't immunize all these stray dogs because it's too costly. The cost is around 500 rupees each. Any idea how many dollars that would be? 500
It's not
rupees?
as much as you think. The real problem is probably not just paying for the vaccination, but getting the vaccine to these infected individuals and recognizing the fact that they actually are or have been bitten by a rabid animal.
Right. So one of these articles is written by a veterinarian who says that the animal birth control plan in India, which was meant to control dog breeding, is a flop. India is the only one in the world that spends more money on maintaining stray dogs than on anti-rabies vaccines for human patients.
Doesn't sound like a good investment, does it?
Problem. It sounds like a problem. But so clearly in many countries, rabies is a huge issue because there are so many animals that aren't immunized. In the U.S., of course, it's not an issue because our domestic pets are all immunized, cats and dogs largely. Most of the rabies in the U.S. is due to wild animal sources.
Right, but it's found in high-density populations, and frequently there are lots of rabid raccoons, and they have the ability to pass that virus on to domestic animals, such as your dogs
Absolutely.
and cats, and that's why it's important to maintain those vaccinations.
Did you maintain your pets' vaccinations?
Absolutely, yeah, every year, even on the one that we didn't let out of the house.
In case it did get out of the house, sure.
Or in case the one that was out of the house came in.
Okay, moving on to herpesviruses, which are viruses that you have worked on for many years in your laboratory. And you continue to do so.
And we continue to do so. When I first came to Columbia University, I was very fascinated by a virus that actually infects predominantly primates as we know it. These are viruses that infect New World monkeys. These are small monkeys, and there were two viruses that were very interesting. One was called herpesvirus saimiri, and the other was called herpesvirus atellus. And I was interested in them because they had been shown to be viruses that were indigenous to the primate population, but were able to transform cells in culture and were perhaps responsible for a form of leukemia and lymphoma in these animals. And I thought it would be a great opportunity to begin to correlate the expression of various virus genes with the development of these lymphomas. And that was back in 1974. And it was a project that I started right out of my postdoctoral fellowship, and I received some funding from the NIH and a local foundation for it. And we began to study these viruses. But that got interrupted by something more interesting.
Which was?
Which was our ability to demonstrate that we could take a gene that had been isolated from herpes simplex virus, and that gene encodes an enzyme called thymidine kinase, and we could introduce that gene into animal cells. And this became a technology which is now widely used in very many different applications for looking at how genes function and for producing proteins in animal cells, proteins that can't be either synthesized chemically or produced in bacterial cells, because the bacterial cells lack enzymes that modify these proteins, that give them either extended life in after injection or actually afford properties to them that they require. So various protein modifications are necessary for activity of these agents, of these.
So this effectively diverted you from Saimiri back into simplex?
That took me out of the Saimiri business back into simplex.
Simplex, of course, being a human virus.
Simplex was also a very interesting virus. It was a virus that I had studied as a postdoctoral fellow, and what had really tweaked my interest at the time, and this was back in early 1970, was the fact that it was a very, very large DNA genome, and it was my thought at the time that I could learn something about how animal cells function by studying this very large genome. I was, of course, very naive.
As we all are.
Yeah. Well, it was a tough problem, and I had my very first graduate student, whose name was Yutaka Nishioka, had come to the laboratory and was interested in working with me on problems with herpes simplex virus. And Yutaka discovered that shortly after infection with this virus, a very interesting thing happened to the cell that was the host for that virus. And that was that it stopped producing its own proteins. Not 100%, but a very large percentage of its own proteins were inhibited. And we wondered what it was that was arresting protein synthesis. And Yutaka and I worked on this problem for about 5 or 6 years, and actually opened up a new field, and that was this field of what happens when a virus infects a cell. How does a virus subjugate the host to its own purposes? And what Yutaka identified was something in the virion itself, that is part of the virus particle, that when it infected the cell, resulted in degradation of host messenger RNA and dissociation of host polyribosomes. And as you well know, Vince, polyribosomes are the machinery that's important for synthesizing proteins. So for a virus to be able to attack this very important regulatory control element of a host, that is how it makes its own proteins, to disassemble it and without destroying the components, laid ground for our understanding about how a virus now begins to make its own proteins in the face of what the host has out there, its own messenger RNAs. So virus mRNAs are seen because the host mRNAs are degraded. And they utilize host polyribosomes that have been disassembled by reassembling them with host messenger RNAs, with virus messenger RNAs. And that became a very interesting field. But we got sidetracked on that one too. And this very interesting discovery with TK really put me out of the virus business for about 6 years while we studied how to put genes into cells. And it's just another example of how interesting viruses are and how they provide key tools for our understanding how cells function and how proteins are made in cells. And so it was a wonderful magical mystery tour, and we had developed a technology with my colleagues Michael Wigler and Richard Axel to put almost any gene into a cell and to be able to ask the questions about what proteins does it make and can we express that protein. And that became a wonderful tool for the pharmacology, for the pharmaceutical industry, and they've used that to produce a wide variety of proteins. But Kenny, let's get back to viruses. And herpes viruses remained a very interesting area of study for the laboratory. While we were doing this research on gene expression and transferring genes into cells, it developed a tool for us to look at individual herpes virus genes. And we were very fascinated by discoveries that had been made in a number of laboratories around the world, particularly first by Hannes and Roisman in Chicago, and subsequently by the groups in Glasgow and Boston, that there were a family of genes that herpes simplex virus, and it turns out most herpes viruses elaborate. In fact, just about all DNA viruses have a phase very early in their life cycle where the first genes that they transcribe and subsequently translate are very intimately involved with regulation of gene expression. So when we talk about virus gene expression, especially amongst DNA viruses, we frequently say that genes are temporally controlled in a coordinate fashion. And by that we mean that a gene has a specific time at which it's turned on and sometimes turned off, and that the product of genes that are made first frequently go back to regulate the subsequent genes, the subsequently expressed genes from a virus. And in work by a series of students, Erwin Gelman and Adele Alcara, we were able to demonstrate that herpes elaborated a series of immediate early gene proteins, two in particular that seem to be very important, one called ICP0, and it's not for nothing that we call it 0, but 0 is a protein that is not obligatorily required by the virus for replication. Yet without it, the virus grows very, very poorly. And we were able to demonstrate that this virus gene product activates transcription from a wide variety of virus and cell genes. That was a very interesting phenomenon, and we, in a group in England, Roger Everett, and a group in Johns Hopkins, headed by Gary Hayward, and another one in Boston, headed by David Knipe, were the first to demonstrate that this gene product was a transcriptional activator. And then this work proceeded to demonstrate that we could use this gene in concert with other virus genes to further augment regulation of virus genes. And that became a very fruitful area for research for a while. It was one that we followed because we were really interested in dissecting how virus genes are regulated, what turns them on and what turns them off. And a little bit later, another cohort of students, Eric Liam and Bob Solomon and postdoctoral fellow Christos Panayotides, began studying another gene that was expressed very, very early in expression. And this turned out to be a gene with another virus gene that has many, many functions, as we see over and over again, where virus genes interact with the host and change the way the host regulates its metabolism and, of course, makes it more available for the virus. And this virus gene product was called ICP27. And it was a small protein of about 63,000 molecular weight that was phosphorylated. And when we looked very carefully at the structure of, or at the hypothetical structure of this protein and its DNA sequence, we realized that it contained regions for nuclear localization, bringing the protein into the nucleus, nuclear export, taking the protein out of the nucleus, and RNA binding motifs. And we began to think, what does this protein do? And I think we were the first ones, but I really don't remember now, demonstrated that this protein shuttled. That is, it moved from the nucleus into the cytoplasm and back into the nucleus. And that was a very interesting observation, and perhaps the most important thing that we learned about this was that this protein had a cargo, and that cargo was virus mRNAs. And subsequent work has demonstrated, and this is work by groups in Britain and on the West Coast, Roz Sandry Golden, has demonstrated this protein has just a whole lot of other functions, and those functions include interacting with the polyadenylation machinery of the cell, and poly-A tracks are things that are added to the end of messenger RNAs are very important for regulating their half-lives and getting them out of the nucleus. So this protein interacts with a wide variety of host-specific proteins and protein machines, those that are involved in export from the nucleus, things that recognize the nucleopore, which is that big hole in the nucleus which allows exit, and for apparatus in the cytoplasm which dissociate the protein, which is a transporter, from its cargo. And you want to do that because if you're bringing in very specific RNAs, you want them to be translated. There's a reason for it. And then, of course, the protein is released, and it's got to get back into the nucleus, and it does that using its nuclear localization signals.
So does ICP4 only bind to viral mRNAs?
So that's not ICP4, that's ICP27.
27, sorry.
So
only viral, or does it also bind host?
It's a very interesting question. It's probable that it sees some host RNAs as well, and I don't think the answer for that is clear. The other thing that is clear is that it doesn't bind all viral RNAs. There are some late RNAs that it sees and some that it doesn't appear to see. The binding has yet to be elucidated how those two things interact. But some of the things that we do know that it binds are factors, proteins used for splicing. And the splicing machinery of the cell that is very important for generating host messenger RNAs is not used very much by most herpes viruses, not just herpes simplex, but the Epstein-Barr virus and the cytomegalovirus and the varicella-zoster viruses. These guys don't have a lot of genes that are spliced. And the recently discovered Kaposi sarcoma virus has a lot of cDNA-like copies of human genes. So the introns have been removed. And it's probable that, well, it's not probable, but in fact it's plausible that this results because many of these viruses have an ICP27-like function. It's one of the most conserved genes in the herpes virus family.
Now, for someone who is not intimately familiar with herpes simplex viruses, why is it important to understand these various ways that you've described by which the virus basically takes over most host functions for its own purposes?
Well, there are a bunch of interesting problems with viruses. One of the most interesting problems is their proclivity for forming latent infections. And by that we mean that the first episode of virus infection usually results in some sort of overt lesion, although not always. The cytomegaloviruses and Epstein-Barr virus primary infections are not generally overt, and you don't see a wound.
But for simplex,
you
usually see a lesion of some
usually
sort.
see an ugly little lesion.
It happens very early in life.
Most people are infected with it in the first 10 years of life. And many children are born with herpes infections, which they acquire either in utero or by when they exit the womb during birth through the birth canal. And those are serious infections, especially with a young child that does not have an active immune system. And they must be treated very aggressively. And when we think about treating infections, we look for targets. And there are some very potent drugs that work well with herpes simplex virus, Epstein-Barr virus, human cytomegalovirus, varicella-zoster virus. And these antiviral drugs target generally the viral thymidine kinase, which is a protein that's unique to the virus and very different from the cell kinases, or the viral DNA polymerases. Polymerases tend to be highly conserved, not just among virus families, but across the spectrum of all organisms. The active sites that are used to add nucleotides to make chains of nucleic acid, be they DNA or RNA, are really very much the same from organism to organism. It's the other proteins that are important for differentiating between host and virus and virus and virus. So those drugs, while efficacious, tend not to have quite the efficacy of the kinase-specific drugs because there are conserved elements, and you have to be careful. And we see that over and over again with HIV.
So if a child acquires a herpes infection at birth and it's very serious, they're treated typically with what kind of antiviral?
They're usually treated with an antiviral called acyclovir or penciclovir, and these are various forms of the same drug that are actively taken up in cells that are infected because the cells have the viral thymidine kinase, and only those cells that have virus take up the drug. So that provides a great therapeutic index. That is, uninfected cells don't incorporate the drug. And the second feature is that these drugs are now used by the viral polymerase. And because they cause chain termination, that is, no new synthesis of virus genomes can occur, they're very effective at limiting the spread of infection if given early enough.
So acyclovir and its derivatives were discovered quite a few years ago.
Long time ago.
Have there been any new anti-herpes antiviral drugs developed? Or
most of the new herpes antiviral drugs have been derivatives
of the original
acyclovir. Yes. Or analog.
Correct me if I'm wrong. I remember a story of a new drug developed against the topoisomerase, or an unwinding protein of the virus, that was very good, but was never brought to market because it simply wasn't better than acyclovir, even though it was a new target and it was quite specific.
So topos have been targets for a number of drug companies for a long time. And interestingly enough, none of those have made it to market, to my knowledge. And I don't know why that is. So the efficacy must not be as good when put into the patient as it is in the test tube. You know, frequently going from the test tube to the patient is a long road of discovery and refinement. So
do companies continue to look for new antivirals against these viruses, or do we think that the acyclovirs are sufficient for what we need?
So we know that the acyclovirs are not sufficient because there are a large number of drug-resistant variants that arise. And they arise because of the unfaithfulness of the polymerase. There isn't quite as much editing function in most virus polymerases as the host has. And that results in a finite number of mutations. And these mutant viruses can escape. They're resistant to the effects. Unless, of course, they're in a cell that has another virus that's making the legitimate kinase. There's not a lot of work in the herpes field at this point.
So if you're infected and you develop resistance to acyclovir, what's done next?
Well, what they'll usually use is one of the variants, famciclovir, penciclovir, and again, there's slight differences, and they're used as second-line drugs.
Now, if you're infected at a young age and your infection is cleared, uneventful infection, presumably now you develop immunity to the virus.
An interesting problem.
Yet later on, if you have some sort of stress, as you know, you develop fever sores, which are reactivation of your original infection. The virus has stayed with you for years. So why is that in the face of having immunity to the original infection, this can occur over and over? And that's correct that it can happen over and over.
There are two elements of what we call recrudescence, the virus coming back. And when I used to lecture to the medical students about herpes virus, I would end with a slide which was a picture of a heart with an arrow that went through it, and it said, "Herpes is forever." And of course, the reason for that is that primary infections always result in latent virus. And the virus goes and it hides in your dorsal root ganglia, which are those nodes along your spine, and it sits there. And it sits there as a big plasmid, a big circle in the nucleus of neurons. And there are some very, very peculiar things that we note about that. And the first is that very little is going on. The virus is not expressing virus specific proteins.
Which is good, because if it were, those cells might be cleared.
So that's an interesting problem, because clearance in the brain is subject to the action of different cells than clearance in the body. And the cells that clear in the brain are astrocytes and microglia and these kinds of guys. And we don't know much about that immune system at all.
And we also understand that the body would rather not clear cells from the brain as readily
right.
as
I think most of us need as many cells as we have.
Yes.
And it's not good to lose too many of them. But the virus has found a way to put multiple genomes in a single cell. So there's not just one copy of the virus genome, but probably as many as 50. And that's a very difficult thing to understand. And the reason that I say that is that if a virus enters a cell and we think that it's going to become latent, we think, and of course this is wrong, that not much is going to happen. And yet we get one virus particle with one genome that appears to result in 50 genomes. So there must be some replication of that virus. And the enigma is that it's doing it in a cell that doesn't replicate by itself. These are neurons. These are cells that are done replicating. So the enzymatic activity that is required to replicate virus as we know it is not available. The other enigma is that as the virus goes through its replication cycle, we say there are 3 phases of virus replication: immediate, early, and late. And the early phase is when DNA is synthesized. And we have always believed, obviously incorrectly, that this order is maintained under all circumstances. But it's not possible for that to occur and still have these virus genomes present in the neurons. Now let's come back to that question you asked a while ago. Nice. And that is, shouldn't immune surveillance help keep these guys down on the farm? And what we know about herpes simplex virus is that the virus is constantly testing the host. And many people have what we call silent infections. And we know that because they transmit virus in the absence of overt lesions. Mm-hmm. So they are shedding virus with no apparent infection.
Where is it shed from typically?
Well, frequently it's most often shed from the sex organs. And we know that partners engaged in sex can transmit the virus without an overt infection. And it's more difficult to monitor in women than in men, but we know from studies done in Seattle by Corey and Holmes that women who are surveyed over repeated periods of time will show that virus has been shed. And so that they are active carriers, and we know that that's also true for men.
And again, in the face of an immune response.
Again, in the face of an immune response. How can that be? Shouldn't there be neutralizing antibody? We know that if you sample 50 people, that between 40 and 45 of them will have antibody that neutralizes virus. So what's the mechanism that allows for transmission of virus in the face of the circulating immune system? Tough question. But we think that what happens is that virus gets reactivated in a small number of neurons in an infected ganglia. I don't think it has to be too many. And that this virus travels down the long ganglia of the nerve and then exits through the same pathway that it enters, the dendrites, those little tiny feeders of your neurons. And then where is it sitting? It's sitting right in the dermis and epidermis, and those are the layers that form the skin. And the immune system doesn't survey so good there.
That's right, nor within the neuron itself.
Right.
So that explains it.
That explains some of it.
Yeah.
But we also think, or we always think, and again, that may or may not be correct, that a cell that's infected with the virus expresses virus proteins on its surface.
Mm-hmm.
And I don't know how carefully that's been studied, but there are now systems where that should be being studied. Certainly the work that Lynn Enquist and his colleagues at Princeton are doing with pseudorabies virus and animal herpes virus, they have the chance to look at where virus glycoproteins, which are important neutralizing epitopes, that is, things that are seen by the immune system and important for neutralizing the virus, do they go down those long neural tracts? Mm-hmm. Are they found in the dendrites? And somebody's got to address that question so that important. We help us to understand that, because it's really a black box.
So you mentioned not too long ago that sexual partners can transmit the infection in the absence of the one knowing that they have any virus at all. Now, if the recipient in that case had been immunized against herpes simplex, would that prevent infection?
So that's a tough one. We know, for example, that there are individuals who are infected with more than one strain of virus, because we can differentiate virus strains using what we call restriction fragment length polymorphisms, which differentiates virus subtype A from B from C. So we know that that can happen. And we know that this happens to people who are immune, that is, have circulating antibody. So the answer is there is evidence that that can happen, although you would think that somebody was protected. The reality is that most people don't acquire type 2 herpes virus, which is the one that it's associated with genital lesions, until later in life. They're more often infected with herpes simplex type 1, which tends to reside above the waist. Mm-hmm.
The reason I ask is because, as you know, there are some efforts to make vaccines against simplex viruses. So I'm wondering what the target for those vaccines, who would the target be for those vaccines, and would they be effective if natural infections don't confer any protection anyway?
That's a very interesting question. It's a very difficult question to ask. So we know, for example, that we can make an effective vaccine against a herpes virus because we have a very effective vaccine against varicella zoster virus. Mm-hmm. And that vaccine is a live attenuated virus. The vaccines that the pharmaceutical industry has been trying to manufacture, for the most part, have been subunit vaccines. And to me, it's unclear what the appropriate target is. Mm-hmm. And I'm not certain that anybody else knows either.
I assume you're not investing in these companies then?
It's not where I'm putting my money. But we could go and digress for a moment and talk about varicella, which is another interesting virus.
Which I recently harbored for some time.
And I'm sorry that we didn't get a chance to sample you in great detail.
So I should tell you that we'll recall this because this is an interesting segue to varicella zoster. Probably 4 or 5 weeks ago, I had an itching feeling on my chest, which developed into a rash, which initially I thought was poison ivy, until I realized I don't get poison ivy. And then I thought, I'll bet it's zoster. And indeed, it spread from one level of my rib around to the back and eventually became a nice rash, which was painful, went away, but still continued to hurt.
And does it still continue to hurt?
Now it's starting to subside.
So we're about 5 weeks out.
So one thing maybe you could explain, we say that the virus comes back in a single dermatome. So what is a dermatome?
So a dermatome is a region of your body that contains nerves that arise in a single ganglion.
Okay. So and those ganglia are where my zoster was since childhood. And the initial infection that I had when I was less than 10 years old was chickenpox.
Right.
It's the same virus. In a kid, it causes chickenpox. In an adult, it's zoster or shingles.
Right, but in an adult, it's a reactivated infection.
Yes.
And chickenpox is the primary infection. And chickenpox is a relatively mild disease in the young. And when we were kids, people used to have chickenpox parties because you wanted your kids to have it so that you got it out of the way, and it was a very seasonal kind of an infection occurring.
Now everyone's immunized against chickenpox, at least in the U.S., right?
Almost everyone, and it was a very long road to immunization. The original vaccine was developed by the Japanese, probably 25 or 30 years ago. And Takahashi took a virus, isolate, passed it in guinea pigs, where the virus replicates very, very poorly. And the virus that came out of that was attenuated. That is, it didn't cause as voracious an infection as the wild-type virus did. And because it was 30 years ago, he was able to put this virus into people and to test it. And the rather remarkable thing was that this virus proved to make an efficacious vaccine. The biggest problem with varicella, though, is that it's extraordinarily difficult to grow. And so unlike herpes simplex virus, where we can isolate beautifully formed cell-free virus at very, very high titers, we can't do that with varicella. In fact, the major manufacturers of vaccine, Merck and Glaxo, and I forgot who does it in Japan, have a great deal of trouble making high titer virus. And therefore, the vaccine is very expensive to produce.
So if this was is a rather benign childhood infection, why was a vaccine developed, and now why are we using it in all children?
So there are really two reasons. Like everything else that goes around in the world, there's an economic reason. And when your child contracts varicella, they're highly infectious. And there's a respiratory phase of the virus, which we believe is the most infectious phase, actually. They get a mild illness, and then a week later they break out all over their body with these pox that are full of infectious virus. And as a result of that, the child really is forced to stay home. Otherwise, it gets spread throughout the immediate population. That frequently means that one of two parents has to be home with them. And if both parents are working, it's an economic burden.
Sure. Okay.
I think a better reason, though, now is that the incidence of shingles, that second occurrence of zoster, seems to be decreased in people who are vaccinated. Now, what we don't know is that if people were originally infected by vaccination, that is, they're originally vaccinated, will these people develop zoster?
Why don't we know that yet?
Well, we're not far enough out. You know, people live a long time. You didn't get your painful lesion until you were 55.
That's right.
Okay, and you were infected when you were under 10,
right.
As was I. And in fact, recently, last year, I got vaccinated for varicella, even though I had had chickenpox. And the reason I did that was that a series of studies demonstrated rather clearly that people immunized, people vaccinated with this vaccine in their early 50s had a 70% or 80% lower incidence of shingles over, I want to say it's a 5 to 10 year period. And that's a big difference. And shingles, as you know, is not a fun thing.
Yes, but in an older person, it shouldn't inhibit any kind of activity. It shouldn't have the economic impact. So.
Well, that's not really true. You were lucky. You only had it across a mild case, across one dermatome. There are people who get it all across their face, which is very disfiguring, and of course causes problems with going out in public because they're embarrassed. There are people who have multiple dermatomes that are inflamed, and there are people who get shingles and are in so much pain. You've talked about what we call post-herpetic neuralgia, which is a very, very difficult to describe syndrome, but what essentially happens is you get this rash across that unique area of your body. Everything heals, everything looks normal, and yet you're in terrible pain. And these people frequently can have this pain for years.
Okay, so it can be debilitating and interfere with work and productivity.
And we don't know the basis for that at all. And that's a very fascinating area of research. And it's another one of these areas of research that combines people with different interests, people who are interested in pain, the physiology of pain, the neurobiology of pain, the interaction of infectious agents that result in this. How does the immune system modulate this? So there's a wide variety of research areas that can come together to try and address a problem such as this.
So the vaccine you received, which was meant to prevent zoster, was that the same vaccine that a child would receive?
Actually not. It's a much higher titer
more virus
prep,
present.
more virus prep, more difficult to make. And again, the vaccine companies have run up against this problem of being able to manufacture really high titer virus. And we know that this is something that they remain interested in.
So in another 20 or 30 years, there will be some very interesting studies ongoing to see if the incidence of zoster is severely reduced in countries like the U.S. where
we should be beginning
right?
to see those studies coming out in Japan in the next 10 years and trying to identify that cohort that received the early vaccinations that did not have chickenpox and whether or not they developed zoster.
So if you have not been immunized, as I was never, and you have a zoster incident, can you take an antiviral to dampen the pain?
Well, what we know is that the antivirals, if taken very early, will help to soothe the pathway of infection.
It won't prevent it, though.
Completely prevent it, because by the time you know that it's happened, you have lots of cells that are infected.
Right.
But they'll keep it from spreading to adjacent cells, and they should dampen the infection, make it less severe. So you should be better off if you start that kind of therapy. You should heal faster and recover faster.
So it's a good idea if you have zoster. You should see a physician and perhaps get a prescription for
you should be running and begging.
Very good.
Absolutely.
Excellent. Okay, so we've talked about herpes simplex viruses a bit, and in the same family of viruses are the varicella zoster. Same pattern, infection initially at a young age. The virus hides in a nerve cell of some kind, and then later on it becomes activated. In the case of simplex, it's more frequently activated by various insults.
So there's an interesting difference between the viruses. And this relates to research that I did in collaboration with Ann Gershon, a pediatrician, who is one of the two major people in the United States, along with a woman by the name of Ann Arvin at Stanford, who are really responsible for pushing the vaccine in this country and getting it through all the clinical trials. And Ann and I looked at ganglia from people who died and had zoster at death. And what we noted was that many ganglia from the cervical, which is all the way up at the top of the spinal cord, all the way down to the lumbar and sacral area, contain virus DNA. So that really recapitulates chickenpox. When you get chickenpox, you are covered from head to toe with virus. And it's easy to understand how every dermatome could become infected. And in fact, that virus is there in all of those dermatomes.
So every dermatome probably has virus, but when you have a zoster attack, only usually one or a few.
Right.
Do you only have one incident of zoster in a lifetime or more?
Unfortunately not.
You can have more.
People tend to have more than one. And that's another mystery, because you would think that second coming of varicella, the zoster, would really boost your immune response. And the reason that I say that that's a real enigma is because if you think about the time span between the initial infection and the subsequent recrudescence, that is the appearance of zoster, you say, it's 40, it's 50 years. That's about the lifetime of what we call T memory cells. All right? And we say, they're gone. All right, so now you're not being surveyed in quite the way that you were before. And maybe that's why you get zoster. But after you've had zoster, you should have boosted that response.
Exactly.
You should be real good at it. And yet people continue to get multiple recurrences. So we don't really understand that. And yet the vaccine is protective for zoster. So there's a bunch of immunology that we don't have a handle on.
So it's safe to say we need to work on these viruses for a long time.
I think if we want to study these guys and address the problem and being able to help people who have infections, that yes, we do need to study them more. And we have been studying that problem. And one of the things that we noted that was very different between herpes simplex, which makes no protein products that we can detect while it's latent, is that zoster makes 4 or 5 protein products. And maybe these protein products are there in a symbiotic way to help keep this guy down on the farm, hidden in your ganglia, and that they provoke the immune system just a little bit. But we don't know that for a fact.
Right. One of the difficulties with these viruses is using animals to study them. It's very difficult to study them in animals, correct?
Zoster grows really only in people. And as you know, when we were studying ganglia here at Columbia in the mid-'90s, we had to have a lot of car accidents to get those victims. But the reality is that we don't have good systems for studying these. We don't have good systems in vivo. We don't have good systems in vitro. And that's really plagued our understanding of establishing latent infection and reactivating from latent infection. With simplex, we have a better model with the mouse, which unfortunately most of the time doesn't reactivate spontaneously, which is what we would like. The rabbit does reactivate spontaneously, and that's given us some information. But we don't have the immunologic tools with the rabbit that we have with the mouse. So you give up something to get something else.
And what about varicella zoster?
Well, we tried for a long time to establish a decent in vitro system. And we've been plagued by our inability to reproducibly establish latent virus. And the other problem with the in vitro systems is that although nerve cells don't actually grow in vivo and divide, you can't keep them in culture forever. So we don't have what we need, which is a reagent which would be latently infected neurons that we could harvest at intervals. We can't propagate them, so we constantly have to make new ones. And we need a better model system. We need an animal model system.
So all of you budding virologists out there, take note. Herpes simplex and varicella zoster viruses need more people to work on them, as long as we can convince our funding agencies to continue supplying the needed capital, of course. Well, I think we can wrap this up today. Thank you for joining us. It was very nice of you. Is there anywhere people can go online to find out more about your work?
Well, actually not.
Not at all?
Not at all.
You don't have a website or?
No, I've never gotten to the point of developing a website. But if you go to the Columbia University microbiology website, you can see a profile of what we do in the laboratory, and there are references to some of the manuscripts that we've published over the years. But maybe it's time to build a website.
Well, we'll put a link in the show notes to what you have, and with the idea that maybe you'll build it up. And today we've only touched on a few of the many more herpes viruses. We talked about herpes simplex, varicella zoster, but of course there's cytomegalovirus, herpes viruses 6 and 7.
And 8.
8, Kaposi sarcoma, Epstein-Barr virus. So perhaps you'll come back another time and we'll talk about those viruses.
We can continue this discussion. I'd love it.
And we'll have Dick Despommier. So thanks for listening. Remember to send your questions and comments to TWiV at twiv.tv. Come back next time. Thank you.
Thank you.